Chapter 8 Cell the Unit of Life. When you look around, you see both living and non living things. You must have wondered and asked yourself what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has. The answer to this is the presence of a basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of one, independent existence, and two, performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which form the tissue of the plant. At about the same time, Theodore Schwann, 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissue, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schleiden and Schwann together formulated the cell theory. This theory, however, did not explain as how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells, omnis cellula e cellula. He modified the hypothesis of Schleiden and Schwann to get the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and products of cells, and all cells arise from pre existing cells. An overview of cell. You have earlier observed cells in an onion peel and a human cheek cell under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material DNA. Cells that have membrane bound nuclei are called eukaryotic, whereas the cells that lack a membrane bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plants and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum (ER), the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are not only found in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, mycoplasmas, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometers in diameter. The nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. The figure 8.1 shows the diagrams of different shapes of the cells. The first one is red blood cells, RBCs, which are also called as erythrocytes. Their shape is round and biconcave. Then white blood cells, amoeboid, columnar epithelial cells, long and narrow, nerve cells, branched and long, a tracheate, which is the part of xylem, it is elongated, mesophyll cells, round and oval. Mesophyll cells are present in the green parts of the plant. These mesophyll cells contain chlorophyll, which help in photosynthesis for making food in plants. Moving on, prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma, and spirillum, spiral. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. This means that in mycoplasma, there is no cell wall surrounding the cell membrane, whereas all the other prokaryotes have this. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles, like the ones in eukaryotes, are found in prokaryotic cells except for ribosomes. That is, no organelles present in prokaryotic cells except for ribosomes. That is, ribosomes are present in prokaryotic cells. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially enfoldings of the cell membrane. Figure 8.2 shows the diagram comparison of eukaryotic cell with other organisms. A typical eukaryotic cell's size ranges from 10 to 20 micrometer. A typical bacteria 1 to 2 micrometer. PPLO, pleuropneumonia-like organism, about 0.1 micrometer. Viruses 0.02 to 0.2 micrometer. Next topic, cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of differences in the cell envelope and the manner in which they respond to the staining procedure developed by Gram-Bis. Those that take up the gram stain are called gram-positive, whereas those that do not take up the gram stain are called gram-negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called slime layer in some, while in others it may be thick and tough called the capsule. Uh, there can be two types of glycocalyx. Slime layer or capsule. Slime layer is a loose sheath, whereas capsule is thick and tough. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacteria from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in the cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion processes, and to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions under the cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extensions from the cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, villi and fimbriae are also surface structures of the bacteria but do not play a role in motility. The villi are elongated tubular structures made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissue. So villi are elongated tubular structures, whereas fimbriae are small bristle-like fibers. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer to 20 nanometer in size and are made of two subunits, 50S and 30S subunits, which means when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into proteins. In prokaryotes, the ribosomes present are of the type 70S, whereas in eukaryotes, they are of the type 80S. Inclusion bodies. Reserved material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granule, synaphysin granule, and glycogen granule. Example of inclusion bodies are phosphate granules, synaphysin granules, and glycogen granules. Gas vacuoles are found in blue-green and purple and green photosynthetic bacteria. Next topic is eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes, which are present in the nucleus and also have a nuclear envelope around the nucleus. All eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell wall, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plants. So basically plants have cell wall, plastids, and a large central vacuole which is not present in animals, and animals have centrioles, which is not present in the plant cells. These are the two diagrams that are given. The first one is plant cell, the second one is animal cell. The markings are important, they can be asked in identifying questions. Let us now look at individual cell organelles to understand their structure and functions. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially human red blood cell RBC erythrocytes, enabled the scientists to deduce the possible structure of plasma membrane. These study shows that cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards the inner part. This ensures that the nonpolar tail of saturated hydrocarbons is protected from the aqueous environment. In addition, the phospholipids membrane also contains cholesterol. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid vary considerably in different cell types. In human beings, the membrane of the erythrocyte, that is the RBC, red blood cells, has approximately 52% protein and 40% lipids. This is an important line. In human beings, the membrane of RBC have 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane, while the integral proteins are partially or totally buried in the membrane. Figure 8.4, fluid mosaic model of plasma membrane. This fluid mosaic model was given by Singer and Nicholson in 1972. Here we can see an integral protein. It can also be called as a transmembrane protein. This is the phospholipid bilayer. The lipids are arranged. Hydrophobic tail spaced inside, whereas the polar heads on the outside. The spiral-looking structure is a peripheral protein, and the structure which is attached on the peripheral protein is a sugar. The yellow dots that are seen are representing cholesterol molecules. An improved model of the structure of cell membrane was proposed by Singer and Nicholson 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower concentration. Water may also move across the membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentrated gradient, that is from lower concentration to the higher concentration. Such a transport is an energy-dependent process in which ATP is utilized, and it is called active transport. Example, the sodium-potassium pump. Next topic is cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of a young plant cell, the primary wall, is capable of growth which gradually diminishes as cell matures, and the secondary wall is formed on the inner towards cell membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their function are coordinated. The endomembrane system include endoplasmic reticulum ER, Golgi complex, lysosomes, and vacuoles. Since the function of the mitochondrial chloroplast and peroxisomes are not coordinated with the above components, these are not considered as part of the endomembrane system. The endoplasmic reticulum ER. Electron microscopic studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum ER. Hence, ER divides the intracellular space into two distinct compartments, that is luminal, which is present inside the ER, and extraluminal cytoplasm compartments. That is, ER divides the cell into two compartments, intracellular space, that is the lumen inside the ER is the luminal compartment, whereas the other portion of the cell is called the extraluminal compartment. The ER often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on the surface is called rough endoplasmic reticulum RER. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum SER. RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SER, that is the smooth endoplasmic reticulum. In figure 8.5 endoplasmic reticulum, this is the nucleus. The envelope consists of nuclear pores. Then it's the network of endoplasmic reticulum. The portion of endoplasmic reticulum which has ribosomes are called endoplasmic reticulum. These green dots represent the ribosomes. The portion of endoplasmic reticulum which does not has ribosome are called smooth endoplasmic reticulum. Second part of the endomembrane system is the Golgi apparatus. Camillo Golgi, 1898, first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae of 0.5 micrometers to 1 micrometer diameter. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cysts or the forming phase. The portion of the Golgi apparatus located in the diagram, which is nearby to the nucleus, is called the convex or the cyst or the forming phase, whereas the portion which is away from the nucleus is called the transphase, concave, or the maturing phase. These structures which you can see, these are flat disc-shaped sacs. These are called cisternae. Figure 8.6 shows the diagram of Golgi apparatus. Concave trans or the maturing phase. The cis and the trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside of the cell. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. This is an important line. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Next part of the endomembrane system is lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrates. Optimally active at the acidic pH, these enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. The last component of the endomembrane system is vacuoles. The vacuoles is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against the concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. That is, the amount of energy that is required by the cell is directly proportional to the number of mitochondria present in the cell. In terms of shape and size, also considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical, having a diameter of 0.2 to 1 micrometer, average 0.5 micrometer, and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane-bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called cristae, singular crista, towards the matrix. The cristae increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP, adenosine triphosphate. Hence, they are called the powerhouses of the cell. Mitochondria are called the powerhouses of the cell. The matrix also possesses single circular DNA molecule, few RNA molecules, ribosomes 70S, and the components required for the synthesis of proteins. The mitochondria divide by fission. Figure 8.7 shows the structure of mitochondrion longitudinal section. This is the outer membrane. This is the inner membrane. As you can see, these two membranes divides the organelle into two compartments, the outer compartment and the inner compartment. The inner compartment consists of matrix. The space between both the membranes is called intermembrane space, and you can see the inner membrane, the enfoldings are called crista. Then the next topic is plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into three types: chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplasts, fat-soluble carotenoid pigments like carotene, xanthophylls, and others are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastids of varied shapes and sizes with stored nutrients. The leucoplasts are of these types: amyloplast, elioplast, and the eleuroplast. The amyloplast stored carbohydrate starch, example potato. Elioplast stored oils and fats, whereas the eleuroplast stored protein. So we can repeat again. Eleuroplast store protein, amyloplast store carbohydrate starch, whereas the elioplast store oils and fats. Majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40 per cell in the mesophyll. One chloroplast is present in the Chlamydomonas green alga, whereas 20 to 40 chloroplasts are present in the mesophyll cell. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stacks like the piles of coins called grana, singular granum, or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids encloses a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosome of the chloroplast are smaller 70s than the cytoplasmic ribosomes 80s. So eukaryotic organisms have 80s ribosomes. Prokaryotic have 70s ribosomes. Also, the eukaryotic organisms have mitochondria and chloroplast, which have ribosomes which are of the type 70s. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Palade, 1953. They are composed of ribonucleic acid, RNA, and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S, while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunit. The two subunits of 80S ribosome are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here S, the Svedberg's constant stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Figure 8.9 shows the ribosomes. The purple one is large subunit, the lighter one is smaller subunit. When these two combine, these produce protein. Figure 8.8, sectional view of chloroplast. Chloroplast is a double membranous organelle. The matrix is called stroma. This is the outer membrane, then inner membrane. Then these are sac-like structures called thylakoids, which contain chloroplast. A stack of these thylakoids is called granum. The connections between these granums are called stroma lamellae. Next topic is cytoskeleton, an elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia singular cilium and flagella singular flagellum are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of eukaryotic flagella. The electron microscope study of the cilium or the flagellum shows that they are covered with plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonomal microtubules is referred to as the 9+2 array. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. Figure 8.10. Section of cilia or flagella showing different parts. A. Electron micrograph. B. Diagrammatic representation of internal structure. A figure shows the actual electron micrograph image of the cilia or flagella, whereas B is the diagrammatic representation. So there are two singlets in the center, central microtubules, whereas there are 9 doublets in the outer side. These are two interconnected ones. The peripheral microtubules, these are called doublets. The outer layer is called plasma membrane. There is a central sheath which surrounds the singlets. The doublets are connected to the central sheath by radial spokes. So since there are 9 doublets, there are 9 radial spokes. The consecutive doublets are connected by interdoublet bridge. Next topic is centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles and a centrosome lie perpendicular to each other, that is 90 degrees, other in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and is called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cells. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. Figure 8.11 Structure of the nucleus. This is the nucleus. The membrane of the nucleus is nuclear membrane. The membrane has nuclear pores for exchange of material with the cytoplasm. The matrix of the nucleus is called nucleoplasm. There is a densely stained structure in the nucleus called as the nucleolus. The interphase nucleus, that is the nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli, singular nucleolus. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer, called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules take place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Remember, paramecium has two nucleus. Some mature cells even lack nucleus, example erythrocytes of many mammals and seed tube cells of vascular plants. Would you consider these cells as living? This is an important point. RBCs do not have nucleus, the mature RBCs, and the seed tube cells of the vascular plants do not have a nucleus. The nuclear matrix or the nucleoplasm containing nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as that it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Remember this point. Nucleolus is the site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins, and also RNA. So chromatin contains DNA, some basic proteins called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46, 23 pairs of chromosomes. You will study the details of DNA packaging in the form of chromosome in class 12. Every chromosome visible only in dividing cells essentially has a primary construction or the centromere on the sides of which disc-shaped structures called kinetochores are present. Centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosomes can be classified into four types. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long, whereas the telocentric chromosomes have a terminal centromere. Figure 8.12 chromosomes with kinetochore. These are two chromatids connected at centromere and this shaped structures called kinetochore. Figure 8.13 types of chromosomes based on the position of centromere. There are four types of chromosomes. Satellite is present at the end of one of the chromatid or both of the chromatid. It is called secondary construction. The primary construction is at the centromere. The first one we can see is a metacentric chromosome, which has centromere in the middle. That is, the two arms on each side of the centromere are equal. Then it's the submetacentric chromosome. The centromere is slightly away from the middle of the chromosome, resulting in one shorter arm and one longer arm. Then the next is acrocentric chromosome. The centromere is close to its end, forming one extremely short and the other one long. Then the last one is telocentric chromosome, which has a terminal centromere. These are the short arms, the upper ones, the the centromere, and then the long arm. Sometimes a few chromosomes have non-staining secondary constructions at a constant location. This gives the appearance of a small fragment called satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Summary. All organisms are made of cells or aggregates of cells. Cells vary in their shape, size, and activities or functions based on the presence or absence of a membrane-bound nucleus and other organelles. Cells, and hence organisms, can be named as eukaryotic and prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates transport of several molecules. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. All the cell organelles perform different but specific functions. Centrosome and centriole form the basal body of cilia and flagella that facilitate locomotion. In animal cells, centrioles also form spindle apparatus during cell division. In animal cells, centrioles also form spindle apparatus during cell division. Nucleus contains nucleoli and chromatin network. It not only controls the activities of organelles, but also plays a major role in heredity. Endoplasmic reticulum contains tubules or cisternae. They are of two types, rough and smooth. Endoplasmic reticulum helps in the transport of substances, synthesis of proteins, lipoproteins, and glycogen. The Golgi body is a membranous organelle composed of flattened sacs. The secretion of cells are packed in them and transported from the cell. Lysosomes are single membrane structure containing enzymes for digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm or are associated with endoplasmic reticulum. Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate. Mitochondria help in oxidative phosphorylation and they help in the generation of ATP. That is why they're called as the powerhouse of the cell. They are bound by double membrane. The outer membrane is smooth and inner one folds into several cristae. Plastids are pigment containing organelles found in plant cells only. In plant cells, chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is the site of light reactions and the stroma of dark reactions. Important line, the grana in the plastids is the site of light reaction, whereas the stroma is the site for dark reactions. The green colored plastids are chloroplasts, which contain chlorophyll, whereas the other colored plastids are chromoplasts, which may contain certain pigments like carotene and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and chromatin material. The cell is the structural and functional unit of life. Thank you. Chapter completed.